Welcome to this first video looking at A-level biology topics, and here I'm looking at simple sugars. On the screen you can see three hexose sugars. These are sugars which have a six-membered ring, and they are on the left-hand side. On the right-hand side is one pentose. This is a five-membered ring. The first hexose shown here is called alpha glucose. The second one is called beta glucose. Alpha glucose and beta glucose are isomers, and you can see the difference here in the position of the OH group. In alpha glucose, the OH group is pointing downwards. In beta glucose, the OH group is pointing upwards. The third sugar here is beta galactose. Now, beta galactose is similar to beta glucose. There is one very important difference. The OH group here and the OH group here are in the opposite orientation. In beta glucose, the OH group points downwards, and in beta galactose, the OH group points upwards. Now, to more precisely describe the groups, we need to number the carbons. So on hexose sugars, we start with CH2OH, and I want to label this carbon 6. The next carbon here is 5, 4, 3, 2, and 1. Similarly, beta glucose and beta galactose will be labeled in the same way, starting with CH2OH. The carbon at this position is carbon number 6. Now, the pentose here is alpha fructose. Now, alpha fructose is a five-membered ring, a pentose sugar, although it does have 6 carbons, and you can see them labeled here. On the slide in front of you, you can see 3 disaccharide molecules. Each disaccharide contains 2 monosaccharides joined together by a glycosidic bond, and that is the C-O-C bond, as we can see here. Now, the first molecule we have is called maltose. It contains 2 glucose units joined together, and we have 2 alpha glucose, as noticed by the OH group pointing downwards, and the O here, which was from the OH group of the first glucose. The second molecule we've got here is sucrose. Sucrose contains alpha glucose, and it also contains beta fructose. Okay. The last molecule we've got is called lactose. Now you can see the bond here is drawn in a very strange way. Now lactose contains galactose, and this is beta galactose. And it can also contain glucose in the alpha or beta form, and you can see here that beta glucose here, but it could as well be alpha glucose. Okay. Now maltose is a sugar that you get when you digest starch using the enzyme amylase. It breaks apart the carbohydrate, the polymer starch, to get 2 sugar units, disaccharides, maltose. Sucrose is the sugar that we get from plants such as sugar cane. So this is the cane sugar that we normally use in cooking. Lactose is an important sugar found in milk. So these are 3 disaccharides Now we're going to look at glycosidic bond formation. Now you may have seen monosaccharides drawn in a simple way as shown here. In this simpler orientation, in this simpler diagram, you can see that we have missed out many of the groups. The only groups we're interested in are the H and the OH on both sides, as these are involved in forming bonds. And you can see here we've got alpha glucose. If I then draw a second alpha glucose, you can see that there are two groups which are together and they can form bonds. And you can see these groups are here. We have an OH here, and we have an OH on the other side. And I'm going to circle these three atoms, because these three atoms can form water molecules. So when we form glycosidic bonds, we add these monosaccharides together, and what we get is a disaccharide, and you can see that the O that I have not circled is now part of this bond. And you can draw it like this. In a condensation reaction, we get water released. So if two monosaccharides form one glycosidic bond, we will have one water released. And we need to apply the N-1 rule. So now I want to discuss the N-1 rule, which can be applied to any molecule which forms a bond and is involved in condensation reactions. So we have two monosaccharides. These are monomers. These react together to form dimers, like the disaccharide glucose, or polymers. Imagine we have a molecule containing 4 glucose units, all bonded to each other linearly, like this, the glycosidic bond. So we're going to have 4 glucose units, and I want to draw them like this. One, two, three, and the final one here. And on the top here, I'm adding the CH2OH, carbon 6, and the associated atoms. So I have 4 glucose units, but I only have 3 glycosidic bonds. So if N is the number of glucose units, the number of glycosidic bonds formed will be N-1. So in this case, we have 3 glycosidic bonds, and we will have 3 waters being released, 3 H2O molecules. Now, if we add water back, in this case, in this case, we have condensation, water is released. If we add water back to this glycosidic bond, we can go in the opposite direction, and this is hydrolysis. And it requires water to be added. Now, you can see that bond formation in any biological molecule is reversible. Here, this involves condensation. So the reverse is going to be hydrolysis, and that's why we can make and break bonds in biological polymers. In this section of the video, I go beyond AS level and A level specifications to go into more detail about these sugars. Now, what is not commonly known is that if you have an alpha form of a sugar, a monosaccharide, then you will have the CH2OH on one carbon and the OH, and this is going to be the OH on carbon 1 or 2 in the opposite direction. Now you can see here that the CH2OH group here and the OH group here are pointing in the same direction. That is beta glucose. You can see the same principle for beta galactose. Here you can see the CH2OH and the OH are pointing in the same direction. For alpha fructose, we're looking at the following. We're looking at the CH2OH group here, carbon number 6, and we're looking at the OH group on the lowest number carbon, carbon 2 in this case. You can see they are in the opposite direction. That's why it is alpha fructose. If we flip these two over, then this molecule will become beta fructose.